It is really a pleasure for me to introduce our first afternoon speaker, Dr. Sara Sawyer, who is an assistant professor in the Department of Microbiology and Genetics at the University of Texas, Austin. If I were to think of a word, I guess, to describe Sara, maybe it would be something along the lines of a paleovirologist. She studies basically the evolutionary arms races that are being waged continually and have been waged for quite some time between viruses and the hosts that they infect. And one of the things that she's really interested in is how these arms races basically influence the emergence of new viruses into the population, and whether or not she can identify successful host strategies that can then be used to try and exploit it to basically prevent new viral attacks. So Sara began this work as a postdoc with Harmeet Malik at the Hutch, where she basically studied molecular evolution of HIV restriction factors. She wasn't always a molecular virologist, though. Sara actually got her bachelor's degree in chemical engineering. And before she got her PhD in genetics, she worked for several years as a drilling engineer in the oil industry, often on offshore rigs in the Gulf of Mexico. I'm not sure whether that's going to make her welcome, per se, in Berkeley. But that said, she's gone back and reinvented herself, right? She left it, saw the light, and entered sort of non-oil related science instead. So that was actually something I learned about her when she was a guest on This Week in Virology, or TWIV, which is my lab's favorite podcast. Dr. Sawyer is the recipient of several awards, including the Andy Kaplan Prize in Retrovirology. She was also named a Presidential Early Career Award, received the Presidential Early Career Award for Scientist and Engineer, and presented one of the prestigious symposium lectures at ASV, or American Society of Virology, meeting last summer. So Sara's going to be talking to us today about species barriers, what keeps animal viruses from infecting humans. Welcome.
Oh, wait a second, I'm not speaking on oil-related topics today? I was unclear. Is this mic working? Yeah? Okay. So thank you so much to Britt and CEND and everyone for inviting me, and thank you to all of you for coming to hear my presentation today. I have a couple old friends in the audience that I want to extend a special thank you for showing up and listening today. All right, so as you know, humans face many different bacterial, viral, and eukaryotic pathogens, and all of these pathogens came to us at some point or another from nature. For instance, HIV-1 was a cross-species transmission from chimpanzees. Africans are regularly infected by Lassa fever virus, which they acquire through contact with local rodent populations. New strains of flu come to us from birds and pigs. And Plasmodium falciparum, the causative agent of malaria, was transmitted to humans from gorillas. So when pathogens switch from one host to another host, this is a process called host switching. And when the new host is humans, we give this a special name, which is zoonosis. So this slide represents our current understanding for how host switching occurs. New diseases arise when a virus of one species infiltrates a second host species, shown here in green. And what typically happens is that you'll have infection, and this may lead to a small local transmission chain, and sometimes this could result in epidemic spread. And we understand this to be a multi-step process where the virus genome is selected for a series of mutations that allows it to do the following things better in the new host species: evade immune detection, replicate in cells, spread between individuals, and even modulate their own pathogenicity. But if you think about this, humans are exposed to millions of viruses every single day, yet zoonosis is relatively rare. So it's really important that we think about how this process begins, because once a virus is infecting a human and it is replicating, it has the potential to mutate, and therefore it has the potential to evolve and to possibly move throughout this pipeline. So I want to illustrate to you how we believe that this process begins, and I'll do that by showing you a small portion of the genome of this imaginary green rodent virus. So as you all know, all genomes, but particularly viral genomes, are constantly experiencing mutation. And we believe that cross-species transmission occurs when a jackpot combination of mutations arise. Okay, I don't know what was happening there, but I apologize for the last 3 minutes. Okay, so we believe that this process begins when a jackpot series of mutations arise that give this virus a foothold into a new species. And we actually have some very specific examples of this. This example comes from one of the most famous zoonoses ever, the cross-species transmission of simian immunodeficiency virus from chimpanzees to humans. Now, what I'm showing you here is the protein sequence of a single viral protein called matrix. We know that this cross-species transmission has occurred several times, giving rise to the major HIV groups that now circulate on Earth. And Paul Sharp and colleagues noticed that each time this transmission had occurred, it was accompanied by the same methionine to arginine mutation at the 30th position in matrix. And further, if you take HIV and passage it back through chimpanzees, this mutation reverts. So what this tells us is that this is a very species-specific mutation in this viral genome that probably reflects an underlying genetic difference between humans and chimpanzees. The problem is that it's very difficult to find mutations like this. And the reason is as follows. By the time we discovered HIV in the 1980s, 40 years had passed since this transmission event had occurred. And during that time, the HIV genome continued to mutate and the SIV genome continued to mutate, making it very difficult for us to identify these magical mutations that accompany key events like host switching. So ultimately, we don't just want to look backwards at what viruses have done in the past. We want to be able to predict, given a particular animal virus that we're worried about, what mutations does that virus need in order to replicate and spread in humans? And that's a very difficult problem. But my lab and several other labs are interested in addressing this. And so we study how animal viruses evolve to replicate in human cells. Like I mentioned, the strategy that most labs take to studying this is let's sequence hundreds or thousands of viral genomes and let's make big giant phylogenetic trees and see if we can study what viruses did in the past as they move between species. My lab has a completely unique approach to this question that actually doesn't involve looking at virus sequences at all. Instead, what we do is we break this question down into two sub-questions. For a given virus, what are the genetic barriers in the human cell? In other words, what's keeping these animal viruses from infecting us right now? And then once we've identified those, it becomes almost trivial to set up experiments to ask what mutations does the virus need to get around these barriers? Okay, so I want to introduce you to one more term, which is host range. Host range describes the species that a particular virus can infect. So as an example, HIV infects humans very well, as we heard in the last talk, and as I'm sure you all know, but it doesn't infect rhesus macaques or almost any other non-human primate. This is the reason why we have no animal model system for studying HIV. Okay, a couple observations. Like I said, a lot of people focus on viral diversity, but I want to pose this thought question to you. If you hold a virus static and don't allow it to mutate, what causes a pattern like this? It's actually differences in these host genomes that cause patterns of host range, because this virus is compatible with some hosts and isn't compatible with others. So host genetic differences first define range and also provide the engine that drives viral evolution as the viruses jump between species. Secondly, I want to point out that, of course, antibodies and other humoral immunity are important to these processes, for some viruses more so than others. But interestingly, many of the patterns that we observe of host range that we observe in nature can be recapitulated in cell culture, where those components of the immune system don't exist. So this tells us that there are strong cellular components of host range as well. Okay, so recently, my lab has been developing a new model for studying host range that I wanted to tell you about. It involves a molecule called the transferrin receptor. This receptor is expressed at the cell surface, and it's encoded by all mammalian genomes. The normal job of this receptor is to bind iron-loaded transferrin in your blood and to uptake iron into the cell. So this receptor regulates iron homeostasis in the cell. Interestingly, obviously this receptor is extremely important, but it's also exploited by several different viral nemesises, which, nemesi, which contact this receptor and trigger their own uptake into the cell. I wish I had great jokes like that first speaker instead of just flaunting my poor grammatical skills for laughs, but okay. So we've developed transferrin receptor as a model for host range, and it turns out this is an incredibly interesting model because the viruses that use it have very different patterns of host range in the wild. The first family of viruses that exploits transferrin receptor are the New World arena viruses. You have these in California, by the way. These viruses infect various rodent species, mostly found in Central and South America. But what's interesting about these viruses is that several of them are zoonotic, especially in South America, and cause these nasty hemorrhagic fevers that are highly lethal in humans that contract them. The second virus that utilizes transferrin receptor is a retrovirus called mouse mammary tumor virus. In contrast to the arena viruses, the host range of MMTV is extremely specific. This virus infects only rodents of the genus Mus, including Mus musculus, the house mouse. And even though our boring brown house mouse here is found all over the world, this virus has never been shown to transmit to other rodent species or to humans. The third virus that uses transferrin receptor is parvovirus of carnivores. Interestingly, there's a very well-described cross-species transmission event that took place of parvovirus in the 1970s, where a virus of cats or a closely related carnivore jumped into dogs. And dogs have remained infected with parvovirus since the 1970s. So work by many people over more than a decade has revealed to us that in the case of all three of these viruses, the ability to interact with the transferrin receptor of a particular host is a strong correlate of host range in nature. This seems to be one of the major molecular compatibilities that has to be in place for an animal to be infected. And in fact, both of the cross-species transmissions illustrated here both revolved around these viruses of evolving compatibility with the transferrin receptor of the new species, either humans or dogs. Okay, so I've been working on the host range of parvovirus infection with Colin Parrish at Cornell University, and we just published that work. So if you're interested in that, you can see that paper. Today I wanted to tell you about these rodent viruses that use transferrin receptor. And of these, we certainly know the most about the relationship of arena viruses with their hosts. And that is largely because of the large amount of work that's been performed by Mike Farzan, Harry Che, and Steve Harrison at Harvard Med, including that this group has produced a co-crystal structure of transferrin receptor in complex with the viral surface protein of one of these zoonotic rodent arena viruses, Machupo-virus. And so we have a co-crystal structure of these two proteins. Now, a co-crystal structure is about the most sophisticated piece of biological information that you could ever hope to have if you're interested in a host-virus interaction. Yet, even with this information, it's extremely difficult to predict what mutations in the virus might allow access to the receptor of a new species. And the reason that it's still difficult is as follows. If you take these two interacting proteins and you peel them apart so that you're looking at the faces that are touching, you'd be looking at these two surfaces right here. And what this co-crystal taught us is that there are 16 amino acid positions in each of these two proteins that are mediating this contact. Now, theoretically, if you could encode each of the 20 possible amino acids at each of these 16 sites, that means that there are 20 to the 16 possible surfaces that could exist on each of these two proteins. Now, that sequence space is very large and makes it difficult for us to predict what's going to happen in nature, where random mutation is the engine that drives things like cross-species transmissions. But I'm here to tell you this isn't as depressing as it seems, my lab has been able to use an evolutionary model to narrow down this sequence space to a small enough number of possibilities that we can actually exhaustively test them in the lab. And I'm going to tell you how we do that, but just in broad brushstrokes, I'm going to tell you that the majority of the positions in proteins like these are important for these interactions just because they're important for the basic structure and property of these interaction surfaces. But there's a small subset of these positions that are what we call specificity residues. And these are positions where mutations finely modulate affinity without affecting the underlying protein. And my lab is sort of in the business of finding these specificity residues, and we've done this in several different host virus systems. So I'm going to tell you about a few of those today. So I want to tell you exactly how we find these specificity residues so that you'll really understand. To find them, we use an old theory from evolutionary biology called the arms race theory. And this theory is so generalizable that I can draw it here in cartoon form because it applies anytime host proteins and virus proteins are interacting. And so in this case, if something encoded by a virus, like for instance, a viral surface protein, contacts something encoded by a host, like for instance, transferrin receptor, then the arms race model applies. If this virus is exploiting this protein, there will be strong selective pressure on the host population for any mutation that arises that's resistant to this interaction. If that occurs, there'll then be strong selective pressure placed back on the virus population for an escape mutation to arise. But now that the infection is back in full force again, there'll be selective pressure back on the host population again. And so the critical thing about the arms race model is that it's a continuous loop. There's no equilibrium here because the interests of the host and the interests of the virus are exactly opposite. The host population is being selected to decrease this interaction, and the viral population is being selected to increase it. So both of these proteins are undergoing positive selection for new mutations that arise that change this relationship to the other, that change their relationship to the other protein. So if we think about the evolution of this host protein, in the absence of a virus, we would expect the protein sequence of most host proteins to remain relatively stable, because these proteins are already evolutionarily refined for the function that they perform in the context in which they perform them. But in the presence of a virus, when the arms race is on, we see accelerated protein evolution, particularly at the interface between these two proteins. And specifically, and this is the whole key to finding the specificity residues, specifically we find the most rapid evolution at the codons that can modulate this interaction without a cost to the underlying proteins. Those are the positions where time and again amino acids will be replaced as this war is playing out, because they change the interaction without affecting the underlying protein. And so for the aficionados out there, we find these residues using a rate of evolution statistic called dn over ds, and we estimate this on sequence alignments using maximum likelihood. But here's an example where we've performed this analysis on the transferrin receptor. So this is a small portion of the protein alignment of transferrin receptor from a bunch of different rodent species, most of which are infected by arenaviruses in the wild. And I'm showing you this to show you how this analysis works. You can see that many of the positions in this protein are perfectly conserved. A couple other positions, like this one right here, have experienced one mutation. But these two positions in yellow are on the evolutionary treadmill, and they're changing so fast that almost every single species we look at encodes a different amino acid at these positions. And our model shows that these are evolving much more rapid than would be expected by random chance. So these aren't just neutrally evolving residues. Okay, so when we analyzed transferrin receptor, we found 6 amino acid positions that were evolving like this. I'm showing you the latter 2 here. We found 6 that look like that. And I was a little bit disappointed when I saw this result, because remember, the arms race model says that a host protein like transferrin receptor should have a very specific binding surface that's evolving rapidly. And these are all spread out along the length of the linear structure. They don't look like a binding surface to me. But one of my coolest days in science in the last couple years was when my postdoc brought me these sites translated onto the transferrin structure. So this is the transferrin receptor is a dimer, so there's a mirror image axis here. So one monomer, the other monomer. And here's what these 6 positions look like when they're put onto the 3-dimensional structure. Now this looks like a binding surface that viruses might contact. And so this was really cool to find. And further, we know from the co-crystal that the first 3 of these residues that are experiencing positive selection lie in the arenavirus binding surface, which is shown here in gray. This was great. This made sense. Everything's starting to add up for an arms race between this host protein and viral protein. But we still had 3 unexplained residues, these last 3 here. They don't lie on the portion of transferrin receptor where these arenaviruses bind. And so I didn't know what to make of those. And it was at this time that I remembered that there was this other rodent virus, MMTV, that also utilizes this receptor. So I remember it was about 2 in the morning, and I was sitting there, and I pulled up this old paper from Susan Ross's lab where they mapped the important components of transferrin receptor for binding this other virus. And they made chimeric molecules, and they found 2 regions, a region from 285 to 296, which incorporates this amino acid, and a region from 569 to 589, which encompasses these 2 amino acids. So I was so excited because what we have here is a second virus binding surface. These 2 regions are now indicated in blue, and you can see that 3 of our sites of selection lie in that second binding phase. So what we have here is we think we have a situation where we have a single very important host housekeeping protein, your iron uptake receptor, that's being antagonized by 2 different viruses on 2 separate surfaces, and that there are 2 separate host virus arms races going on here, and that a very small subset of the residues, 3 in each of these binding surfaces, are acutely modulating what's able to interact with what. Okay, so we needed to test this. So again, our hypothesis is that residues under positive selection in transferrin receptor control interactions with viruses without affecting receptor function. Okay, so to test this, we had to set up a host range assay. So what we did is we cloned the transferrin receptor from all of these various exotic rodent species from Central and South America that get these viruses. And we used retroviral vectors to make stable cell lines expressing those receptors. And we put a flag tag on the extracellular domain so that we could use a labeled flag antibody to measure receptor incorporation in live cells during the course of our experiment. We then wanted to infect these cells with arenaviruses and MMTV, the rodent viruses that use this receptor. The one problem here is that arenaviruses are BSL-4 pathogens. In fact, arenaviruses are one of the 7 pathogens in the world included on the CDC Category A biothreats list. So this is not an easy family of viruses to work with. But luckily, since my lab is predominantly an HIV lab, we were able to quickly sort of change courses. And we used pseudoviruses where we pseudotyped the surface proteins of these various rodent viruses onto retroviral vectors. So these are basically dummy viruses that from the outside look exactly like the rodent viruses we're trying to test. And these viruses deliver a payload of a GFP gene. So we know that the viruses have entered through a particular receptor when the cells turn green. Okay. So I now want to convince you that this assay recapitulates the host range that we observe in nature. If you're not familiar with rodent phylogeny, which you may not be, here are the 2 major families of rodents. We've got the Muridae rodents, which includes our boring friend, the house mouse, who is the only host of MMTV. And then the Chrysotati rodents include these various Central and South American rodent species which harbor these arenaviruses, several of which are zoonotic. Okay, so what I'm showing you here is we've expressed these different receptors, the transferrin of the house mouse, which is our brown mouse, and then these fancier ones from the Chrysotati clade. And you can see that just like we see in nature, MMTV is extremely specific for getting into cells through the transferrin receptor of its natural host. It doesn't get into cells through any other receptor, including human. But Machupo virus, this was the one in the co-crystal. This gets into cells best through the transferrin receptor of its rodent host. It can use one other rodent transferrin receptor. This is its close relative here. And it can also use the human transferrin receptor. And as I told you, this is a major reason why this is a zoonotic virus that infects and kills people. but it doesn't get through all receptors. And so while this is very interesting, we wanted to give ourselves sort of the ultimate challenge, which was this virus that had very, very specific host range, and see if we could modulate that through the information that we gleaned from our evolutionary analysis. Another reason that we were more challenged with the MMTV situation is that there's no co-crystal in this situation, and this is not known to jump species, so we imagine there's a pretty large genetic barrier here. It's not easily escaping around whatever the problem is. Okay, so here's my very silly drawing of this beautiful butterfly structure here that's supposed to be transferrin receptor. And so what we've done here is we've taken, this just recapitulates known data. We've taken cells that don't express transferrin receptor, MMTV doesn't get in, but if you give MMTV its transferrin receptor from its favorite host, it gets into cells very well. We then did an experiment where we changed transferrin receptor is 760 amino acids long, just so you know. We changed just the 3 amino acids in this binding surface that are under positive selection to match the amino acids encoded by one of these rodents that doesn't support uptake. And when we do that, we completely destroy the ability of viruses to get into these cells. And I'm not going to show you all the controls, but I'll just tell you these mutations do not affect receptor expression. Remember, we've got that live cell assay to monitor that. Or they also do not affect cellular entry of arenaviruses, the other viruses that enter through the upper part of this receptor, contact the upper part of the receptor. Okay, so this explains why mutations at specificity residues are selected for, because they offer a protective effect against virus entry. Just 3 mutations. If mus musculus had these 3 amino acids instead of the 3 that it does, MMTV probably wouldn't exist at all, since this is its only host. Okay, we then wanted to know if we could do a gain of function mutation where we took the receptor of a non-host species, introduced the 3 amino acids of the host species, and sure enough, we get a significant gain in entry through this. And this was really we thought this was quite impressive given how strict this virus is for its own receptor. So to summarize this data, we produced both gain of function and loss of function swaps in host range of transferrin receptor using only positive selection to identify residues that determine or dictate this range. Now, I wanted to tell you a little bit more about the host functions of transferrin receptor. I told you that this regulates iron homeostasis, and there are 2 human proteins that are known to bind transferrin receptor in this process. Iron-loaded transferrin, we have co-crystals of both. Iron-loaded transferrin binds through this central channel, and the negative regulator of iron uptake, HFE, this is the hemochromatosis protein, also binds through that same region. You can see that these 2 important host interactions lie nowhere near where these evolutionary battles are taking place. But just to check that this evolution wasn't coming at a cost to these normal host functions, we also did an experiment where we took the 2 rodent receptors, including the swapped versions, and tested for the binding of iron-loaded transferrin. And you can see that while these 2 rodent species do bind transferrin slightly different, the mutations in them don't really change those levels. So we think this evolutionary model, in a way, has found mutations that separate the virus binding function of these receptors from the iron loading functions. And separation of function mutants are always useful. It helps us understand the trade-offs between these processes. Okay, so I've been telling you about these evolutionary arms races. All selection in nature starts by a random mutation or a SNP that appears in one individual. And so imagine that we have these different rodent populations, and one of them gets a random mutation. And let's say that this random mutation affords this rodent a very big advantage, or even a small advantage. Like, for instance, let's say that it prohibits viruses from entering through transferrin receptor. If that happens, then there will be selection on this SNP. Another way to say that is that rodents that don't have it will die with bias. And so you'll be left with a higher frequency of SNPs in this population. And it's this process playing out time and time again that has led to the rapid evolution that we observe at these sites and these codons. And so we wondered if maybe this type of evolution could now be affecting human transferrin receptor. And the reason we wondered that is because several of these viruses, like I mentioned, do infect humans. And I wanted to tell you about one of these. It's called Hunin virus, and it causes Argentine hemorrhagic fever. This virus is transmitted by the laucha, which is a rodent found in Argentina. And it's a seasonal disease that infects agricultural workers, specifically during the corn harvesting season. And the reason is because these farmers drive these big mechanical corn harvesters, and they stir up all that dried rodent urine and feces and aerosolize it and breathe it in and contract this virus and get hemorrhagic fever. And people who get this disease die 20% of the time. So this is, and you know, hemorrhagic fever is like you bleed from your eyeballs and stuff, so nobody wants to die this way. But I was just testing if you're awake. Okay. Okay, so because this Argentine hemorrhagic fever is a huge deal in the area where people are interfacing with these rodents, obviously a lot of work has been put into studying these. And there's actually a facility here in Junín province in Argentina where a vaccine is made. And this is a picture looking down over the balcony in this facility at these large vats where the Candid #1 attenuated live vaccine strain of Junín virus is grown so that local farmers can be vaccinated against this virus. Also at this facility, they store big aliquots of serum from survivors. So if some farmer is bad and doesn't go get vaccinated and gets this illness, they can infuse them with a big load of serum from survivors. And just for fun, I wanted to show you the horse that serves as the lawnmower of this facility. Really? Okay. So I told you we're now talking about SNPs, and maybe now that these viruses are emerging into humans, are we all equally susceptible? Or maybe this arms race is starting to affect us as well. So we knew where these arenaviruses touch this receptor, so we searched human SNP databases, and we found a human SNP at position 212. It's a leucine in most people, but some people encode a valine at that position. And we cloned these 2 variant transferrin receptors in 3 different cell lines. In all cases, the wild type allele is on top and the rare allele is on the bottom. They differ by only 1 amino acid. And you can see that the receptor that has this single amino acid is less susceptible to virus entry in all 3 cases. And that's even true when this receptor is expressed at higher level. So that means that it looks like this SNP could have a protective effect and could protect some people from either contracting this disease or from the severity of it once it occurs. And so humans harboring this SNP would be predicted to better survive infection. So we learned, by studying these rodent reservoirs and the evolutionary dynamics that are going on there, now we can extrapolate this into human and understand the biology of how that might translate to zoonotic disease. So what about the virus side of this arms race? I told you that the arms race is this back and forth war between the host and the virus. And so I've told you about all this rapid evolution on the host receptor. Is the virus responding in turn? Well, for this study, we switched back over to Machupo, not Junín anymore. We switched back to Machupo, and we were able to acquire a bunch of Machupo virus sequences from, Machupo is a disease in Bolivia, and we acquired samples from both humans and the Vesper mouse, which is the local rodent that transmits this virus to humans. And we performed positive selection analysis on this little virus protein here. And so here's a picture. Sorry, I switched colors. It's brown here, it's green here. This is the glycoprotein ball at the end of the virus spike. And so we found this in blue is showing the residues that coordinate interaction with the receptor. We got that from the co-crystal. And in red are the residues that are experiencing this recurrent positive selection, that have super high variation and are being selected. You can see that they all fall on or flanking this receptor binding site, and we don't find any residues under positive selection on the other side of this glycoprotein that faces the virus. Now, with the virus, it's a little bit more difficult because, of course, there's immune selection going on, too. So this signature that we're seeing could be a composite of both of those selective pressures. OK, so I'm now going to tell you, I'm going to try to extend this tool and tell you that we can actually use positive selection analysis to test theories about disease emergence. So this story involves SARS, which emerged as a new human disease in 2002. And we think, yeah, nobody laughed this time, OK. I guess the movie's getting a little old, right? Okay, so anyway, we think SARS emerged as a new human disease in 2002, and we think that a coronavirus from bats infected palm civets that then infected humans. And this is a really straightforward transmission model, and there's a lot of evidence for it, like the fact that bats and palm civets both have viruses that look incredibly similar to SARS. But for the last decade since this disease outbreak occurred, there's been one huge problem with this model. And it has led to all sorts of cockamamie alternate hypotheses that involve viral recombination between multiple disease reservoirs. The problem is this. All the coronaviruses related to SARS that have been isolated from the civet or from human all utilize this cell surface receptor called ACE2 to enter into cells. But no coronavirus ever isolated from bats has been shown to use ACE2. And further, the viruses that have been isolated from bats have a deletion in the ACE2 binding region, as inferred from homology with these other viruses. And so this has caused a real problem. But of course, we don't have exhaustive sampling of every bat virus on the planet. So maybe there are SARS-like coronaviruses in bats that use ACE2, we just haven't found them. But this has been a standing problem. And even though SARS is gone, many people feel it's still important that we understand what happened here, because we can sit around and worry about bird flu all we want, and by the way, we should be worrying about bird flu, but this virus came out of nowhere, and nobody was worrying about this before it happened, at least as far as I know. So it's important that we understand exactly what took place. So we addressed this question by looking at the evolution of the ACE2 receptor in bats. So these are all the different bats that we looked at. And when we analyzed the ACE2 receptor, we found a bunch of residues that have experienced positive selection. So this tells us that something is driving the rapid evolution of ACE2 in bats. We then mapped these residues onto the human ACE2 receptor because there's a beautiful co-crystal with the SARS spike protein. And lo and behold, if these signatures of positive selection don't exactly fit on this binding footprint of SARS onto this ACE2 protein. When we saw this, we had to ask ourselves, if there's no virus in bats that's using ACE2, why is this molecule under positive selection? And secondly, why is it under positive selection right in the binding surface with ACE2? With SARS. So we did several other things in this paper that I don't have time to go into that helped us sort of convincingly make the argument that positive selection reflects evolution in response to a virus that's very similar to SARS. There were some other alternative hypotheses, but this really looks like something SARS-specific. And we concluded that ACE2 utilization by coronaviruses in bats is ancient. So ancient that it's been able to leave an evolutionary signature in this gene in bats, but that we just haven't found these bat viruses yet. So our model supported the straightforward transmission chain. And excitingly, we presented this work last summer at the American Society for Virology meeting. And the very next day, a group from China reported that they had finally found the ACE2 utilizing virus in bats. And so this kind of completed the circle on the SARS transmission model, and so now I think there's a lot of evidence that suggests exactly what happened. Okay, so with the final minutes I have, I'm just going to tell you one more tiny story about one more problem of host range. This is my favorite virus, HIV, and as I mentioned, it has a pretty distinct host range as well. It turns out that for HIV, the major genetic determinant of host range we've learned over the research that's been conducted in the last decade turns out to be a family of proteins called HIV restriction factors. For HIV, it's not the receptor, it's these proteins really that determine host range. So HIV restriction factors, in case you're not clear on what that means, these are a kind of newly appreciated family of immunity proteins that are expressed inside of your cells and detect viruses that are trying to replicate in there. And one of these is called TRIM5 alpha. And TRIM5 alpha is an E3 ubiquitin ligase that has these four protein domains. TRIM5 alpha is a pattern recognition receptor, or PRR, for retroviral capsids. So this is a picture of a retrovirus entering cells, and retroviruses uncoat and expose this capsid core, shown here in purple and here in white. And TRIM5 alpha is a cytoplasmic protein, not shown here, but shown in color here. And this protein detects the pattern of this weird-looking shape along with the specific protein sequence of the capsid protein and forms this hexameric lattice all around this capsid core. And through torsional strain, it cracks it like an egg and blocks the viral life cycle. So cool, right? Okay, so here's a little cartoon of that. TRIM5 is the cytoplasmic protein. It detects this capsid core, cracks it, and then the virus can't go on to replicate anymore. So the good news is that rhesus macaque TRIM5 works great against HIV, and in fact, most primate versions do, which is why we can't infect any of them, we now understand, in the primate facility. But the human version is totally lousy and restricts nothing, as far as we can tell. Okay. So here is another instance where we have a host protein, TRIM5, interacting with a virus protein, capsid. And you may say, but wait a second, HIV is only 80 years old. Surely there's not evolutionary arms races playing out yet. But I'll remind you that HIV is really just the primate lentivirus of the day. Primates in Africa have been struggling with simian immunodeficiency virus, their version of this virus, for millions of years. And many African primates harbor their own flavor of this virus. And several times, these viruses have leaked out into the human population, giving rise to the HIV variants that we now have. And so in order to study this protein, TRIM5 alpha, I can't sequence it from bats or rodents. I have to go to the reservoir for this virus, which is primates. And so here are just a few fun pictures of me chasing primate samples, which is what I spend a fair amount of time doing. A lot of them we get from cell culture banks, but sometimes we do sampling things. This is a picture of an owl monkey, one of the only nocturnal monkeys. This is a picture of 2 ring-tailed lemurs that I took at a lemur preserve. This is what I'm doing when I'm not drilling for oil, by the way. This is a silver gibbon that I met in Indonesia. This is a Japanese snow macaque that I met in Japan. And this is a postdoc collecting poop from this mongoose lemur back here. I'm outside taking pictures, by the way. So we collect a lot of material from primates. We mostly get blood and skin biopsies. And at this point, I have probably 100 to 200 primate biomaterial samples in my freezer. And we grow these cell lines. So in my tissue culture room, we might have bonobo skin cells growing or TT monkey blood cells growing. And so you never know. So we create cDNA libraries and study these genes from those materials. So for TRIM5 alpha, we sequence the gene. I'm going to speed up just a little bit here. We sequenced the gene from this family of primates. By the way, you are a hominoid up here with the great apes and the lesser apes, and you've got these are your cousins. Okay. So in this case, we analyzed the evolution of this TRIM5 protein, and we found 5 residues under positive selection. You can see how crazily variable they are. But in this case, unlike transferrin receptor, these behaved nicely, and they fell in a tight little patch at the beginning of the B30.2 domain. I found this as a postdoc, and when I saw this, I said, this has to be the virus interacting domain of this restriction factor, because remember, positive selection is supposed to accumulate right at the interaction interface between these proteins. And so I tested that as follows. I took the human TRIM5, which doesn't work against HIV. That's why we get infected. And the rhesus macaque, which does. That's why they don't get infected. One of the reasons they don't get infected. And I just made a simple swap of that 11 amino acid patch containing those residues and almost completely reversed the ability of this restriction factor to recognize HIV. We infected cells expressing these variants. And so now, actually, my refs got cut off, but I've worked on TRIM5 for a lot of years now, and I just show this so I can close with 2 final slides showing where we've gone with this story. We now know, after hundreds of papers after this finding, which we made in 2005, that this little region constitutes a single loop extending out of TRIM5 alpha. And this is the little tongue that's feeling around for those crazy capsid eggs. And if it finds one that it likes, it suctions onto there and makes its lattice and cracks the egg. So this is the viral recognition loop we now appreciate. And I just want to point out, we found it just using a computer analysis. And we were the first paper to show that this was the region that mattered for that. And we only made exactly 1 chimera. That's actually how it happened, not just how we wrote it in the paper. Okay, so I just want to show the current status. TRIMs are my very favorite gene of all, so I have to end just by showing you our latest paper that we just published. We found 30 new TRIM genes in the human genome. Now, all of you actually, we already knew you had 70 TRIM genes. What I mean by a TRIM gene are genes that encode proteins with these domains and that can be aligned to TRIM5. So we found 30 new genes on chromosome 11. I told you already had 70, so who cares, right? But these are kind of special for a certain reason. These were not named or annotated. We found these bioinformatically, but they almost all have complete open reading frames and are predicted to encode a protein that looks just like this. And we've now generated transcript evidence for most of them. There are 3 segments of these genes on human chromosomes, on human chromosome 11, and we think that this segment right here is what we termed a gene fountain. And we learned that by doing primate comparative genomics. You can see that if you look at various primate genomes, this segment 1 is old and was acquired back here before these primates diverged. This segment here is a segmental duplication of this region. We proved that through some other methods, synteny and stuff like that. And this occurred before the split of humans and chimpanzees. Only they have it. And then there's a second copy of this segment 1 right next door that happened just on the human lineage. And further, so we think that this gene fountain, if these are restriction factor genes, we think that this gene fountain is sprinkling primate genomes with new versions of these genes that can be evolutionarily refined for new viral targets and, in the typical way that your immune system loves to do, create novel substrates for the development of new specificities against new pathogens. So this is so volatile that it's even active in this room right here. We used an assay for copy number variation, and we showed that even this thing is still sprinkling genomes in the human population, and some humans have extra copies of this segment compared to other humans. So I wanted to show you this because this is a really different paradigm for species tropism. What if just the dosage of these genes in human compared to rhesus macaque might create a barrier for the transmission of viruses between these species? So we don't know if that's true yet. Okay, just to summarize, we started with observations of host range in nature, and we've determined how genetic divergence between species dictates resistance or susceptibility. And in all 3 of these cases, we've been able to do that to the amino acid resolution. But if you'll recall, that was only our first step, unfortunately. I guess I have to work on this a little longer. But what we really want to do is test, once we know those genetic barriers, what mutations viruses need to get around them. But I've shown you that transferrin receptor, ACE2, and TRIM5 alpha sequence divergence all create these barriers, maybe even TRIM gene copy number. And what I wanted to tell you is that this work has already started. Colin Parrish has shown us in exquisite detail how parvoviruses evolve to get around the transferrin incompatibilities in new species. And Welkin Johnson at Boston College has shown us how simian immunodeficiency virus evolves to get around incompatible TRIM5 alphas, or rather compatible TRIM5 alphas. And so while this seems like a tremendous amount of work, I hope I have convinced you that this is at least a rational approach for how to try to answer this very difficult question of predicting what viruses are going to do when they encounter a novel species. And with that, I'll just thank the people that did the work, my postdoc, Ann Dimogenes, an MD PhD student in my lab, Diane Liu. They both are supported by fellowships. My postdoc, Kyudong Han, is now assistant professor in Korea. These are the 3 people who did the work I showed today. We also collaborated with Mike Farzan, Harry Che, and Jonathan Abraham at Harvard Medical School, who supplied us with reagents and great advice on all of these projects, actually. They identified several of the receptors I talked about in the first place, so good collaborators to have. Thank you so much for coming.
Questions?
Great talk. I actually have 2 questions. The first is, in your cell culture system under positive selection, what can you say in the host about effects on DNA repair mechanisms like nucleotide excision repair? Is it diminished under those positive selective pressures? And the second question is, there's a company in town that's essentially knocking out the CCR5 co-receptor to establish a stable hematopoietic cell population as an approach against HIV. Would you recommend sort of, in high-risk populations, stably introducing, TRIM5, like from rhesus macaques or something to, have an anti-HIV.
So I'll start with the second question, and just to repeat it, it's now in clinical trials, the idea to treat HIV by taking out somebody's hematopoietic stem cells or T cells and knocking out the CCR5 co-receptor and then giving those T cells back to them. It's called a functional cure because you don't cure HIV, but you cure AIDS because you create a protected reservoir of T cells that will stave off immune collapse and the onset of AIDS. He's asking me, in light of what I showed, would it be good to put a primate version into that locus? It's been done in 5 papers. Sorry, I'm not trying to make you look bad there, but yeah, it's been done in 5 papers. Come on, So, so my little hybrid human rhesus TRIM5 molecule has now been being put into this locus because it's almost all human, which is good for rejection issues, but it's not. But it works against HIV. And in fact, I'm a co-author on a paper with Matt Porteous where we've just introduced this idea of restriction factor stacking. So we used zinc finger nucleases to deliver 3 potent restriction factors into the CCR5 locus. It's like genetic heart. There are 4 genetic barriers in these T cells that HIV would have to simultaneously evolve around, and that's not just an idea specific to us. Other people have been thinking of that too. So yeah, we're trying to do that. And your first question I didn't quite understand. Could you restate?
Oh, I'm just wondering if somehow DNA repair is inhibited in the context of really strong positive selection.
DNA repair.
Mechanism. So you get, mutation is it's easier to mutations arise more quickly, and so in your cell culture.
Okay, so you're throwing me a little bit off guard because it's like you have insider information. The whole other side of my lab works on DNA repair, so I was like, I didn't talk about that today, but okay. So I think the question is, and correct me if I'm wrong, the question is there, it turns out that DNA repair is a really central hotspot for the battle between hosts and viruses. Because viruses come in as either single-stranded RNA, double-stranded RNA, single-stranded DNA, or double-stranded DNA, but they don't have telomeres, so they all look like crazy foreign objects that the DNA repair pathway tries to shut down, and absolutely viruses are fighting back. And so we've got a story on that. It's a little bit preliminary, but did I answer your question?
Yeah.
Okay, thanks.
For one more.
Very clear presentation, so clear that I can actually ask a question. Basically, I'm wondering about sort of the reverse of this issue, which I think is probably best described by thinking back to the bat-SARS relationship. If the bats have basically a binding site for the SARS virus, why doesn't the SARS virus infect the bats?
So that's a great question. And it's really a central issue. When viruses, you guys have all heard the term attenuation. In the olden days well, we still do actually, we make alive attenuated viruses by putting them in a foreign species. And we think about attenuation as crippling. But really what you're doing is making the virus stronger for that new species, which often comes at the cost for its replication in the first species. And so we think that's exactly what happened with SARS. It refined itself for civets and humans at the cost of sort of fluidly going back and forth. And thank goodness that it's not just a giant mixing pot of viruses out there which can infect everything. That does seem to be a common theme, although we don't know a lot about the evolutionary trade-offs.
So is that same logic true for the prior mouse lack of cross-reactivity?
We haven't shown that, but I would love to show that, yeah.
I'm sorry to cut off questions, but let's thank you for a really great talk.